On this podcast, you'll find interviews with high-performing, successful individuals in life sciences. On a weekly basis, we cover their proven methods, principles, strategies, and mindsets to implement new technologies that scale to meet the needs of people in our world.
Welcome to Life Science Success Podcast. For those of you who don't know me, my name is Don, and I'm a consultant in life sciences, and I help companies overall focus in on their business operations around strategy, people, process, and technology. So with that, in this episode, my guest is Dr. Thomas Rademacher. Professor Rademacher is a PhD and MD. He co-founded Emergex and served as CEO since the company's formation. He's Emeritus Professor of Molecular Medicine at the University College of London and a serial entrepreneur. So with that, welcome.
Well, I'm glad to be here today.
Yeah, it's great to have you. And I'm really looking forward to this because, I talk to a lot of people, I feel like, in this field, and there are a couple of topics that I feel like that your profile really sort of highlights. One is this sort of researcher to serial entrepreneur, right? It's the, kind of the career journey that a lot of people take. And I feel like it's something that we'll have a great conversation about. So thanks a lot for being here.
I'll only add that journey has been cyclic. I think they say there's a famous graph of publications that academics do, and it should be a sine wave, because you can't think up things 24 hours a day, 7 days a week. So if you look at a lot of the great experimentalists, they have a public lots of papers and a few years in the doldrums and lots of other. And mine was very similar in the sense I was an academic, then into entrepreneurial, then back academic, whatever it is. In other words, the famous thing, never be in the same job for more than 10 years. I think I've sort of stuck to that, I have to say. So.
Yeah. And I do feel like it's a popular journey as well. So
Sure.
Would you mind just telling the listeners just a little bit about yourself?
Well, I've got I started out obviously an MD PhD, and that was when I think they really meant something. I have to say, that's when your PhD had to be a real PhD. Now we have lots of MD PhD programs, but I got a good solid PhD in biochemistry at the Institute for Enzyme Research at the University of Wisconsin Madison, which I think is in my case was probably one of the best degrees best things I've ever done, because it really was, let's put it this way, hardcore biochemistry. And it's amazing how often over your career you have to go back to fundamentals such as understanding water structure or things like that, which of course is no longer taught. So I think I was very fortunate in having a very, very strong background, and then also and then applying that throughout my career to basically clinical research, which is probably what I have been in right since the beginning.
It's interesting that you say that the path of academics has changed, and in terms of the structure of the material that you learn has changed as well. Those fundamentals are absolutely important. I would imagine that they're most important whenever you're looking at a potential failure point or a potential challenge that you could be running into in any of the companies that you're working on as well.
Yeah, no, I agree. And of course, there was many years with me and my colleagues were, of course, complaining that nobody was being really taught the hardcore facts. When we started out, we literally had to sit, every reagent you used, you had to make. Now, research is, if you can't buy it, you don't do it. I mean, that's actually quite true. In other words, I can't buy it, so therefore I can't do the experiment. So I go back to the old days when you had to make your own. You had to distill your own toluene. You had to everything had to be done by hand. But what I'm saying is, in the overall process then of a whole career, it's amazing how many times those initial skills do come back, because not everything can be bought. nowadays. In other words, a Sigma catalog does not contain the total glossary of chemicals and things you need. And chemistry is real, and you need a lot of you need to know those basic skills of how to do these things. When we were at Oxford, for example, the we were now you have questions about the Glycobiology Institute, but really we were one of the few people that did a lot of the initial sequencing and actually in developing the field of glycobiology, because one of the reagents you needed was a hydrazine, and you needed to distill anhydrous hydrazine, which of course is rocket fuel. And there's very few people in the world that are brave enough to distill anhydrous hydrazine in a laboratory. And so we had a very, very sophisticated setup with all kinds of gas trains and everything else around. But that was the reagent you could do it. So it sort of gave you a little bit of jump on the competition when there was, well, you had to do it. And of course, I learned how to do that when I spent some time in the great laboratories of Akira Kabata in Japan, where they also worked out a lot of the initial things that were used in that early years of glycobiology. So.
Very interesting. Yeah, and I do look forward to just diving a little bit more into that as well. So whenever I looked at your profile, you have over 200 publications. You have 50 patents. Do any of these achievements really stand out to you in terms of, things that were most pivotal in your career, and why?
Yeah, I think there was a whole series of things that uh, I put it this way. I was very lucky, and I walked into something that was absolutely just at that point in happening. So when I went to Oxford in 1980, the head of the department there had just recently actually had just received the Nobel Prize for the immunoglobulin antibody. We forget these things. It was only at that around 1980 and things we're talking about. What does an antibody look like? And of course, he had described the antibody Porter along with Edelman, and they got the Nobel Prize for that. But then the race was on to actually do the first X-ray crystallography structure of the antibody molecule. And this was being done by a group in Germany. Diessenhofer and Hofer eventually got the Nobel Prize for the structure. But anyway, they took the X-ray structure of this thing, but they couldn't solve it. And the reason for that was it had a little bit of a nuisance in the center of it. It had some blurred, what's called non-defined density in the center. And of course, this was carbohydrate.
Mm.
And the antibody molecule is unique amongst all proteins in the body in that it has the sugars on the inside of the molecule. You see all these pictures of proteins and things with all these sugars hanging out. They're all hanging on the outside. But the antibody molecule, it's on the inside. And it's a very, very interesting point. But the point is, they didn't know the structure of this carbohydrate or whatever it was in the center. And that made solving the actual original X ray structure for the antibody molecule very difficult. So we got interested in this in Oxford because I was working along with Porter's group and everything else. So we decided, well, we'll go out and try and determine what's in the inside of the antibody molecule. So there was one group that only one group in the world at that time that really had any type of technology to do that, and that was the group Akira Kabata's group in Japan. So and he was notorious for never having Westerners in his group. So I sort of showed up at his door one day, literally, and he sort of took me to heart too a little bit. And anyway, I learned how to make some of the basics of what he was doing, and brought some of that technology back to Oxford. And then, of course, threw the entire weight of that institution behind how to do this. And eventually we got to the point we thought we could do it, and we got a hold of the actual crystal that the original X-ray was done on, because we had to know what was actually in the crystal, we had to use what was called radioactive sequencing. But since the crystal isn't very much of something, we had to get Amersham to make us a special radioactive pot of borohydrate that was literally melting the glass, I have to tell you. And we succeeded in sequencing the carbohydrate inside of that, and that allowed for the first X-ray structure of the antibody molecule. And when you look in any textbook in the world and you see the sugar on the inside of that molecule, which is two N-link complex biantennary sugars, that was us. That was us.
Very nice.
So that allowed for that, but that had horrendous, huge downstream implications, because about the same time, monoclonal antibodies had just been discovered by Kohler and Milstein. So the monoclonal antibodies, of course, they have a sugar in them. Okay, that became a problem, because if you made the antibody in a mouse, it ended up with mouse sugars in it. Or if you made it in another species, it made it, and our body was rejecting all these monoclonals because they had the foreign sugar in. This is all the problems behind giving xenotransplants today. So we had to, therefore, I'm going back to one of my greatest achievements, which is a very interesting thing. They needed a cell line or something that would make a monoclonal containing a human sugar. Now, of course, you could spend your lifetime trying to find such a thing. And we did find one by chance, and that's the famous Chinese hamster ovary cells, the CHO cells. So I'm the CHO man. Let's put it that way. The reason recombinant proteins today and monoclonals all the early days were made in CHO cells is because a Chinese hamster ovary cell line produces human sugars, and that allowed the monoclonal field to begin. Without that, they couldn't have done it. In other words, they didn't have the technologies for CRISPRs and everything else. So you can see knowing a little sugar created that, and that came true for the recombinant protein. We were the first people to file a patent on tissue plasminogen activator, which was the original Genentech. That set Genentech. That was Genentech's very second, I think, or their biggest product, and that was still used today. Everybody who gets a blood clot or has a heart attack gets a shot of Activase, which is a tissue plasminogen activator. And again, there it was made initially in mouse cells or whatever it is, and they needed to be able to make it so it was compatible to be injected. And that was the fundamentals of glycobiology. So I look at it as we would not have recombinant proteins today, nor monoclonals, if I had not gone to Japan to figure out how to bloody sequence the original this thing. But those were two massive implications of the field of glycobiology because of this species incompatibility issues and whatever it is. And now, of course, a lot of things are made differently than they were then. But those, to me, if you want to look at it, were two, when I think about it, major achievements that I'm very proud of that have changed probably the whole history of medicine since the 1980s. Something as simple as a CHO cell making human sugars. So anyway.
That is really interesting. I mean, and I could tell you, I mean, already in this episode, I'm learning an awful lot, in terms of, foundational chemistry as well as foundational elements that really helped monoclonal antibodies, make it into the medicines that we have today. The interesting part for me is, a bit of my history. I worked for General Electric for a number of years, and they acquired Amersham Pharmacia, as you recall. And I remember that, my first days on the ground in the company, there was all of this conversation about monoclonal antibodies and the impact it was going to have. And of course it was, a decade later, all of a sudden you start to see the impact. But I mean, it was, a good number of years from that moment that I can recall. And I could only imagine kind of your history with this from
Yeah.
sequencing sugar.
Yeah, it's because then even there, then it became, or if you want to look at it that way, is that so once we learned how to sequence the sugars, of course, that was the foundations of glycobiology. And my God, it is difficult. I have to say, the things that I mean, the early days when you had no idea what you're dealing with now. I mean, it was so basically we created an entire field, and one of my greatest achievements, my colleague who I worked with all those years, Raymond Dweck, we always say our greatest achievement is that we have several words in the Oxford English Dictionary, and that is glycobiology, glycoform, and glycotype. So we made up these words. I have to say, what we literally did. We originally called ourselves the Oxford Oligosaccharide Group. That's a big mouthful, I have to say.
I'm glad it didn't stay that.
So we were arrogant enough to write an annual reviews of biochemistry and title it Glycobiology. And that has been recognized as the first time that word had been used in that sense. And of course, now there's departments of glycobiology all over the world. There's professors of glycobiology, and it's a major field. But the next implication of all of that was that we had the sort of the idea that there was people that for a long time had a suspicion that antibodies or immune complexes, whatever, were involved in rheumatoid arthritis.
Mm.
Nobody in those days really knew much about rheumatoid arthritis. In fact, we still don't know the fundamental causes of it. But anyway, with the Japanese colleagues, then we published I think one of the most, I consider one of the greatest papers in glycobiology, showing that the people who had rheumatoid arthritis had what's called bad sugars. What I mean by, so basically they produce an antibody molecule in which the sugars sort of didn't sit in the center. They're supposed to be inside, if you can
Mm-hmm.
imagine, and they
Sure.
sort of flipped out. And when they flipped out,
Mm.
okay, they became, they set off the complement cascade, they set off inflammation, or whatever it is. And so it evolved from that is of course that, and it still holds today, that the inflammation in your joints or whatever it is caused by a abnormally glycosylated form of the antibody molecule. But that worked its way into the monoclonals because that is not what you wanted to make your monoclonal with. In other words, you not only wanted human sugars, but you didn't want the bad guys. Let's put it that way. So therefore, in this day, without getting into the details, every antibody molecule is designed to be what's called fully glycosylated. It's very interesting when you think about it. The sugars on the inside of the antibody molecule, why are they inside? It's because they're bacteria-like. So, in fact, every antibody molecule harbors elements that would be recognized by the immune system as bacteria. That's why they're hidden. And that's why they
Mm-hmm.
got to stay hidden. But it was a structural requirement. But if they flip out, then your body thinks this is a piece of group A strep.
Mm.
So it was a beautiful bit of engineering by the human body to do this, but it could go wrong. So bad guys never sit on the outside; they can only sit inside the molecule. So anyway, from that, the evolved in that, and then going into that, if you ask questions about entrepreneurship at that point, this whole business came to the attention of the Monsanto Corporation. And of course, they were so we had formed they really would sort of form the Glycobiology Institute, and they became very interested, very, very farsight. They had a very big part of the Searle organization at that time. They became very interested in glycobiology. They set up an in-house glycobiology unit in St. Louis, etc. And they effectively built the building and funded the formation of the Glycobiology Institute at the University of Oxford. And the spin out of that was a company called Oxford Glycosciences PLC. That was the very first biotech spin out of Oxford University. They
Huh.
never had one before. And got to remember, universities did not do these things in the early 1980s. This was, in other words, there was massive opposition to this by the governing body and all of our colleagues, whatever it is. In other words, to actually take your academic research and turn it into a biotech. And it took us, I don't know how many thousands of pieces of paper and everything else to create. But this was the first biotech spin out of Oxford. And of course, this was just after Genentech and Amgen and Biogen. So this is the very foundations of the whole biotech industry. Anyway, that became a full-fledged company. Eventually went on the London stock market. Eventually, it was bought up by big pharma, as things are. But that still are still drugs developed by that company that are used today. For example, for Gaucher's disease and other things like that. So that's how I got involved in entrepreneurial. In that, basically, Monsanto came along, and then they said, "Well, why don't we try and do something special with this technology?" So that's what led to that. So that's how I got involved with that, and then I went back to I never worked for that company. I founded it and was a consultant, but I then went back to more academic climbs in terms of this sigmoidal part of one's career, I have to say. So
Very
that's way
nice.
that, but that did lead to the first. I think one of the main, and I think that still holds the record as well as one of the largest. Even in there, it was over $1 billion float in those days. I have to
Wow.
say it was a thing. But I do remember, and I have to say, I keep a lot of mementos. So obviously, with the Monsanto Corporation coming in, and by the way, they were great people at that time, very different. And they're now owned by Bayer, but they had great people, great science. They came up with just phenomenal stuff, whatever it is. But we were in those days not allowed to keep our patents. In other words, in
Ah,
fact,
yep.
you couldn't, you didn't get royalties, you get nothing. You just handed it over. Okay,
Right. I'm
so
familiar
I
with that, given
uh,
where I come from. So
all
yes.
right. So anyway, I have kept my check. I have a check cut by the Monsanto Corporation assigning the rights to tissue plasminogen activator, which of course went on to become the largest selling drug, I think, one in the world for a long time, recombinant genetic, for $10. Now I've never cashed it, but I kept the $10 check, and I still have it in the drawer.
That's
That
a nice memento. It's funny because I have things like that as well that I've hung on to throughout my career. And even though they're probably not important to anybody else, they're important to me for a variety of reasons. And it's great
yeah
that you got to hang on to that as well. So tell me a
yeah
little bit about Emergex. What is it that you're working on? What is the company, built and
yeah.
around, and what is it that you're trying to bring to market?
Well, what we're trying to bring to market is basically there is the within immunology there's what's called the holy grail of immunology, which every card carrying immunologist knows that if you're dealing with viral based infections, you got to activate the cellular immune response. Basically, you got to kill the factory. Okay, that's the holy grail. Everybody knows that. Forget antibodies or anything. Everybody knows in real life that's what you have to do. And so, but everybody, but the problem has always been is how do you achieve that? In other words, how do you instruct the immune system to go ahead and kill the viral infected cells? Now, we've only achieved it by chance with the live attenuated vaccines. They're really the only ones that have ever been able to do this. So yellow fever, polio, measles, mumps, whatever. You get one shot, that's it for life. You don't need a booster every whatever. That's it. In other words, and what you've got is good old cellular immunity. And the trick here is called the word live, because you then have to get into the definition of what is a live vaccine. And what I mean by that is if I would take yellow fever vaccine and I vaccinate myself, I'll generate a cellular immune response. If I take exactly that same vaccine and I just shine a little UV on it or a little heat or something like that, inject it under my skin, it doesn't work. So the body somehow recognizes some component in this virus. And of course, there's arguments, is a virus alive? It doesn't metabolize, it doesn't do anything. But yet the body recognizes something that it says, this is a danger, this is alive. And you can do it by upsetting some of the proteins if you heat them, or you can do it by upsetting some of the RNA, whatever it is. But it's arguable. In other words, what the definition. So therefore, the trick has always been is how do you make a vaccine that mimics the question live without being live. I'm
Right.
just trying to put it in a very
Right. No. Yeah.
interesting point of view. Okay, because if you could make a non-living live vaccine, you've solved the problem. Because all those other vaccines were discovered by chance. Yellow fever, 1922. Some guys passaged the yellow fever virus. I think it's 222 times. Eventually says, maybe it's attenuated, and tested it on a bunch of people. You can't do any of that. Those things anymore. And it's like, for example, how would you ever test a live Ebola vaccine? I mean, it just isn't going to happen. So therefore, that's really been it's really been the challenge, is how do you mimic the what's called the contextual. I'm not saying within contextual. That's a bad word to use. I know right now, especially if you're a president of various universities. It is true in the immune system that basically whatever you inject has to be recognized in a contextual basis, and that contextual basis is it's alive. So we didn't really. So there was three things that allowed this to be solved, and three pieces of technology, which is only very evolved in the last several years. Okay, and the first one is high resolution mass spectrometry. Really high resolution mass spectrometry. I mean, really, really, really. And the reason for that is when a virus infects a cell, within 20 to 30 seconds, that cell is already marked for death. It's marked for death because the virus that goes in, as soon as it's any of its RNA or whatever it is, produces anything. Before it produces intact proteins, it produces small peptides. And these things substitute on our HLA system, which recognizes us as self. So all of a sudden, that cell is non-self. It's foreign. As far as the body's concerned, it's foreign. So within 20 to 30 seconds, that thing is going to be nailed eventually. Okay, it's going to be nailed. So the question then came, and that happens in every single viral infection. So the question comes is what is that code? What's those first things that appear on the surface of a viral infected cell? Now, why do we want to know the first things? It's very obvious. A virus, usually one virus infects a cell. They don't come in as a group. They usually come in as one. And what happens is that it takes them time to make more friends. That's the way I usually try to say it. So this cell will eventually explode. If it's an RNA virus, the cell will explode, producing all kinds of virions. Okay, and that is really what transmission is. It's not what goes through the air. That's what's called bottlenecking. You have to get the virology right. So transmission is when the cell explodes in your nose from cell A, and it produces 100 billion friends, and they go out and spread to another 100 billion cells, etc., whatever it is. Now, if you can kill the viral infected cell before it makes any friends, and it takes about 18 to 20 hours to make all the bricks and the building blocks to produce a virus. It's a lot of components. It's got to make all those things, and then it's got to assemble them. So for about the first 18 to 20 hours, this thing can be killed off without producing any progeny. And that's what's called the eclipse period. And that's what's called abortive infection. And that's what's happening in you probably several hundred times a day, because you've got a good old immune system against, you're breathing in rhino, you're breathing in tons and tons of things right now. And what's happening is that one virus is getting in, and your T cells are killing that one viral infected cell off before it can produce any progeny. That's how natural immunity works.
Mm-hmm.
So if you therefore can know what that signal is, that's the first thing. So the problem comes is that you've got about the surface of our cell defines us as self. I'm different than you, and we can't transplant between each other. So the few virus signals that appear are buried within 2 billion possible self-peptides. So that's the needle in the haystack. And that's why I said massively impressive mass spectroscopy was required to fish out these very, very early peptide that derive from the viruses. And that's what happens in a natural infection. So the first thing we did is we discovered how to do that. So we know all of the viral signatures from flu virus. We know the viral signatures from dengue. We know those from yellow fever. We know those from Zika. And it doesn't change in people. It's the same. You got the same set of things. So the first thing is we now know what are the targets. Okay. Now, how are you going to vaccinate somebody against those targets? Well, the easiest way to explain this is if I get a mosquito bite in my arm, okay,
Sure.
with dengue, I get a mosquito bite. It's whoa, sorry. If I get a mosquito bite with dengue in my arm, it's nowhere near my lymph nodes. Okay, the immunology occurs in my draining lymph nodes. But the infection is at the site of the mosquito bite. That's where
Mm,
dengue infects it. The mosquito comes in, it actually spits into you, is what it does. That attracts some myeloid cells. They're the ones that the dengue then infects. So the dengue infection is in the skin, and it basically remains within cells the entire time. There's never a viremic phase, or something like that. So there's never a point where an antibody would actually do anything against it because it's never free in solution. It just goes from cell to cell to cell. Anyway, the point is that information, therefore, has to travel from the site of the viral infection into my lymph nodes. And so there is actually what I talked about, that little peptide signal, actually in a real infection is transported in my lymph up to the lymph node. There's actual physical coding, if you want to look at it that way. I mean, people don't realize, but how it gets there, and there's a mechanism by which that peptide moves from the site of the infection into my lymph node. Once it gets up there, it talks to all those antigen, dendritic cells, whatever it is, and the body then miraculously will generate a T cell army on its own without anything else, which will go and kill off that dengue thing. So we had to then, second of all, develop a delivery system. And delivery system is very, very interesting because what you're doing is trying to do a lymphatic vaccination. In fact, if you think about that, you're vaccinating into the lymph. And second of all, the codes are peptides. Now, a peptide has got a lifespan of about 10 seconds in your blood or your lymph because proteases will eat it for lunch.
sure.
Proteases are there to get rid of every little thing. So the codes we're delivering are lunch to all the proteases, if you want to look at it that way. They want to kill them all off. So you therefore say, well, I want to get these codes up to the lymph nodes. I could put it in a vesicle. Now, as you know, the mRNA vaccines are in a vesicle. Now, here we get into good old biochemistry physics, because what's the smallest bubble that you can make, the smallest vesicle? And it turns out it's about 50 nanometers in size because of curvature issues. You can't make a bubble smaller than with lipids, etc., but smaller than 50 nanometers. If I put something 50 nanometers into my skin, it goes nowhere. My skin is a heavy collagen thatch. And the flaps to my my lymphatics are about 20 nanometers. So it goes nowhere. That's why you could put HIV under your skin and not get infected. It can't go anywhere. It just sits there. So we couldn't encapsulate. So that meant you couldn't encapsulate. So that meant we had to put it on some type of particle, which could move into the lymph that was small enough to go. But that means you had to have the old peptides on the outside. So what I had started a company called Mighty Tech Pharma, which is currently it was on NASDAQ and AIM. And when we did that, that was the world's first nanomedicine company. So I started actually made up the word nanomedicine, I have to say. They didn't call it that in the early days, but we did with the Spanish Research Council. And at the time, we discovered a very interesting particle, and it's actually, I know these guys got the old Nobel Prize for quantum dots, but actually we did it first, I have to say. We discovered a particle which was called a quantum-confined gold cluster, and it is an artificial atom. And it's 102 atoms of gold. It's a very, very strange beast. Okay, it doesn't act like gold. It's non-metallic. You name it. It's funny. Anyway, the point about it is that this particular particle, if you want to call it that, because it's really so small. It's 102 atoms. It's about the size of 10 sugar bits. It creates. It has a quantum field around it. This is absolutely true. It has a quantum field called a Debye field around it, emanating from it. So if I took something fluorescent and came near this particle, the fluorescence would quench. It sees the field. If I try to have a melt, I've tried to freeze this particle, you have a melt zone. Structure of water, we talked about. Structure of
Sure.
water became
Right.
very important.
Hmm.
So it disrupts the structure of water, which means enzymes don't work around the surface of this particle. This is a force field particle, believe it or not, extending out to about 5 to 7 nanometers. And it's on that particle we put our peptide codes. So these things go in. They're what's called stealth. They don't bind to proteins because obviously they don't stick. Nothing can get to them. There's a
Sure.
force field around it, which is
Hmm.
really pretty weird. So in actual fact, that is actually the delivery system that we have been using in human beings. These are called quantum-confined clusters, in which they create a force field that protects the peptide. Now, then you'd say, well, that's never going to go in anything because it's stealth, it's invisible. Because these were called invisible particles. They used to call them ghost particles. Some people, because whatever it is. Now it turns out there's a peculiarity of our immune system that the immune system recognizes these stealth particles. Now, don't ask me why, but they do. Okay, so
Yeah.
if you take all the blood, you take all your PBMCs in your blood and you throw our vaccine in there, they go to exactly the right cells, the dendritic cells and the antigen presenting cells that you want. We don't know why, but they do. They recognize nothingness. That's the way I've tried to describe it. I mean, it's bizarre, but that's actually the way it works. So when these things are injected in, they go up, they don't bind to proteins, they don't get stuck on anything, they don't bind to the endothelial cells. I mean, it's just like, just like the body isn't there, they're walking through walls, but when they get to the lymphoid tissue, the correct antigen presenting cells take them up, and that code is then presented just like it would be in a natural infection, and the immune system gets activated just like in a natural infection. So the third piece that was required was if I'm injecting something in my skin, how am I going to get vaccination in my lungs?
Mm.
Okay, it's a sort of a practical point here. In other words, okay, that sounds great for skin immunization, but how do I immunize? How do I get immunization in the lung? And it turned out a trick was evolved. Turns out if you vaccinate in what's called the epidermal space of the skin, now you've got the you got the original, you got the surface, and then
Sure.
you got the epidermis, and then the dermis. And the epidermis is a very unusual place to vaccinate, but it's got, excuse me, a very unusual compartment. There's actually fluid flow in this compartment. Some people call it the nth organ or whatever it is, and it's the highest density of T cells and Kupffer cells in the entire body. And you can imagine your skin is there as the first barrier to all of this crap that we're getting in every day. So, how do you do that? Because you can't possibly use a syringe to inject into the epidermal layer of the skin. Because if you go into the dermal layer, it doesn't work.
Mm.
Micro, Microneedle technology came along. That was developed. Microneedle technology is a bed of nails, if you want to look at it that way, where you can inject into the epidermis. And we knew this, and other companies now and other academic groups have confirmed that if you vaccinate into the epidermis, you get systemic immunity.
Huh.
So those
That's interesting.
cells that you do in the epidermis in your skin will go out and populate the. So if you vaccinate against, as we did with the COVID vaccine, if you vaccinate against COVID in the skin, you get COVID anti-COVID T cells in your lungs. They
Sure.
go there.
Hmm.
So three major issues solved the holy grail problem, I call it. One was mass spectrometry, learn the code. Two, nanotechnology, how to deliver, and microneedle technology, how to deliver and get systemic immunization. So the base of these vaccines, therefore, are very straightforward and simple. If I would right now vaccinate you against dengue, okay, what I would do is activate your dengue army.
Hmm.
Okay, you are. Most people don't realize that you are born with a set of T cells already. You are up to the age of 7. The dice kept on being thrown, and then about the age of 7, when your thymus involutes, you make no more. So you do not you don't respond to a viral antigen by making a new type of T cell. In other words, you are only given a single throw of the dice. And so if you don't have any dengue T cells by the age of 7, you're screwed. Basically, you've had it. Okay, and so basically that we call it the throw of the dice. So these guys are already there. Okay, and it's by pure chance we produce about 10 to the 11th of these, and the body sort of throws the dice and says, I hope that's everything I'm going to meet in my lifetime. Okay,
By 7 years old, yeah.
The antibodies are different. You can make new specificities all the time.
Sure.
So the antibody system is completely different. Here, you cannot make a new T cell specificity. Now, it gets confusing because we call these naive T cells, but they're only naive because they haven't been turned on, but they
Hmm.
already have specificity. So if we go in there and they see they see the signal, they say, I think there's been a dengue infection going on because we've tricked the immune system. They will automatically pre-programmed multiply and divide every hour. They increase by you make a couple up to 1 or 2% of all the T cells in your body will become anti-dengue within about 24 hours of getting one of these vaccines. And then they slow down and they become memory T cells, and they'll be with you the rest of your life, pretty much next 25 years. And that's how it works. So therefore, what we do therefore is prime you. So when I said the T cell army comes along to kill off this single virus infected cell, in a natural infection, it takes about 8 days to create the T cell army. So it's not there to begin with. Okay, so your other systems, the innate immune system, all this other stuff there's a war going on. And eventually, of course, you do start feeling sick, but then the T cell system clicks in around day 8 or 9 and you kill off the infected cell. But by then it's spread. It's been, in other words, you can see that. On the other hand, if you already have the army there at day 0, when there's only
Mm.
one bloody infected cell, it kills off that one cell because that's what happens in you once you have natural immunity. Once you now have the army. That's why you don't get people are constantly being re-exposed to polio all the time. It's in the environment, it's everywhere else. But one of those things come in, you got the army, you kill it off. So T cell priming basically is doing nothing other than turning on that T cell army prior to the infection. And you get infected, it's there on day 0, goodbye. And that is the holy grail, really, of all vaccinology and immunology. But this is nothing new to immunology. It's just the mechanisms by which we can achieve this. And as I said, these were really technological advances. If you want to look at it that way, they're instrumentation, mass spec, nanoparticles, and microneedle technology allowed us to implement that. On the other hand, you've now got that T cell army. Now, why is that T cell army actually quite important? Because by creating the right T cell army. Now, this is kind of funny to think about. You can't become a ball of T cells.
Mm.
Okay. Now, if you think about that all
Sure.
the time, you're constantly being bombarded. And if we told you every time you came up against something, you're going to create 1 or 2% of your T cells in your blood as you become against flu or against that, whatever it is, you'd become a ball of T cells eventually. So, what happens? The body counts cells, believe it or not, and it sort of fixes the total number of T cells. So therefore, if I create an army of T cells against flu, something's got to give. And so you start knocking off other T cells.
Sure.
Okay. And it's called shelf space. It's a wonderful, wonderful concept.
Mm.
Okay. So the problem with that is if you create T cells what are called off target, and that's what viruses love to do. I'm going to create a T cell that's off target. I don't express it during my eclipse phase, and therefore, who cares? The problem with that is of course you end up depressing your total immune system because you're getting rid of the good guys. For example, it's known one of the young people realize the main reason we give measles vaccine is to prevent this from happening.
Mm.
Measles is a very, very vigorous vaccine. If you catch measles naturally, it produces a huge T cell response and actually wipes out your chickenpox response that you
Ah.
had. Okay, so if you see what I mean, so therefore we give people measles vaccine really to keep them from not losing all their other childhood immunity, and that's actually what happens if they catch natural measles. So it's this shelf space thing, but nevertheless, somebody did the calculation. The total T cells in your body, the immune system, if it was all in one spot, would be about the size of a pineapple. And that's about it, it would be an organ that size. But the other thing to remember is the T cells we're talking about are not in your blood. The ones in your blood are useless. They're just a remnant. 99.9% of all your T cells, immune cells, are in the tissues. That's where they live. And they're at all the entry points. In other words, they're going around. If they're in the blood, it's too late because you got infected somewhere else. So this is a difficulty in clinical trials of these things, is because what you can measure in the blood is not relevant to the vaccination. If you see what I mean,
Yeah.
unless you're going to go in there and do lymph node biopsies or something like that. So
I
it's a
do have I
challenge.
I
Yeah.
do have some questions about your so you're going to JP Morgan. In the trip to JP Morgan, what are you hoping to bring to JP Morgan as well?
Well, actual fact is we're not, because of a large number of things. We're not going to be we're not going to be at J.P. Morgan this year.
Okay.
So I mean, it's been a traditional haunt of mine many years. But we're right at this point in a huge, as you know, we've got lots of clinical trials going on and things like that. And it's best sometimes to be at the base, because you lose a good week and a half when you go to
Mm,
that
yeah, sure.
to that
It's a good
conference.
place to connect with a lot of people I know as well.
Yes,
But
yeah. Oh, absolutely, that's right.
and it does sound like, yeah, I mean, you're working on some really exciting things at Emergex. I do want to, focus a little bit on you as well, though. I have three questions that are standard questions for every guest. What inspires you?
Um, I think the uh, what inspires me is, as I have to say, is that I actually was thinking about that when I know you were going to ask that question. Is I think it inspires me is I think some of the things I have done in my career uh, uh, really were, I have to say, I had a huge opportunity to be in groundbreaking. I should say, for example, the carbohydrates. When we had to, when we were at Oxford, we co-developed two-dimensional NMR,
Mm,
which is now with the Oxford enzyme group. There was, the first NMR, high resolution Fourier transfer NMR, was all developed, 10 feet from my lab. And so all of the mathematics and everything else was also done to solve this problem. But I look back at all the things that we were able to solve, and I think there is somewhat of a legacy that what we did is in those days and even now has will have generational impact. And what I mean by that is I knew a lot of academics who would, for the sake of research grants, always play it safe. They'll just
mm,
do, in other words, I've done cytokine X, now I'll do cytokine Y, now I'll do cytokine B, whatever it is. We were always on the cutting edge. I've got a great paper that I love. It was on the biochemistry of leaves, abscission, why leaves fall off trees. That's one of my favorite. I did it with the Department of Plant Pathology, whatever it is. I mean, I thought that was. I mean, you don't think about it, but it turns out it's carbohydrate mediated. I have to say, is that. But I think I've done some pretty strange things. We did on these things and studied some very strange businesses. But that inspires me. I think that what I did in my career is really making it. It really has made a difference so far, and I think the nanoparticle, or these quantum clusters that we've got. I mean, this is a whole new ball game of delivery. And in other words, I'm not saying I'm fruitcake enough to believe that we're dealing with lots of quantum effects in the body, but we probably are. I mean, there's things that we can do with these things that just don't make sense, if you know what I mean. So I think that is in other words, I'm not saying they're tunneling themselves around, but they're doing something anyway.
So, I mean, if I were to summarize, right, I mean, I think you're just your natural sense of, inquisitiveness and your ongoing, level of sort of, reaching out, for that next answer, sounds like it inspires you an awful lot. The next question is, what concerns you?
Oh, that's easy. Revolving doors. Revolving doors is the biggest thing that concerns me. And if you want to look at the recent COVID issues or whatever it is, revolving doors between health authorities, big pharma, regulators, and all of that. I think that has gotten very, very incestuous. I think it's completely wrong. I mean, my old days of dealing with FDA and everything else, I mean, there was like a war between big pharma and the FDA and the CDC. Now, you can't, they're just a gigantic a blur. Um, so, I mean, I think that is a real worry. We see that all the time. And even in our vaccine business, we see, I see it every day, regulators playing favorites with big pharma and things like that. It's even with what the stuff that we've got. It's battles to get in the doors and to get past regulators and that, because it's all, money is talking right now, and I think something has got to dramatically change, and that is so alien to me now, what I am seeing there. And of course, we all know that. I mean, can you differentiate between the CDC, NIH, FDA, and Pfizer? I don't know. To me, they're all, they're all one now, and I don't think that is good, because they will push a technology at the expense, of other technologies, and that's how advances are made. And of course, the venture capital community is all part of that. And in other words, so that's things I have changed. When we did, interesting, when we did Oxford Glycosciences, I think we joked we had a Series F funding round. Most people won't believe there is such a thing as a Series F, but that just shows you how in those days you could continuously build up a portfolio of institutional investors, etc. Now, who all wanted to get in on the ground floor things. Now, as you know, you try and do a Series A, B, or B, C. They say, "Well, find me a lead investor." Nobody wants to do the work
Sure,
either.
sure.
Find me somebody else to do the lead, and then I will do it. Nobody wants to do the due diligence and the work. So that's really what concerns me, because I know of a lot of really phenomenal things that I've seen just fall by the side, because there's just no way that they are going to be commercialized. And of course, these university business collaboration people. I mean, they need to get back to academia more versus thinking they can, run a pharma company from a academic lab.
Uh-huh.
So,
And the last question, what excites you?
what excites me is, I think it's sort of like in those previous answers. I think the what excites me here is the current technology that we're doing here. Of course, there's a very slightly interesting nuance to all of this. I think it was in the mid 1990s. I think it was, with a group in Pennsylvania, was called Syntara. It was actually the first cancer vaccine company ever formed, as far as I know. It was closed down.
Hmm.
Nobody, absolutely nobody, believed that you could treat cancer with immunology, cancer vaccines, whatever it is. In other words, it absolutely nobody. You could not get a single pharma investor, anybody involved in it whatsoever. And the and now, of course, as you see, that is the big issue now. We all know now that if we can deal with the cancer environment, there's no reason why a cancer cell, which is technically being seen as foreign due to various reasons, should not be eliminated. So what excites me now is, of course, while we're currently focusing on the infectious disease, I think that this potentially will make a huge impact in some very in some cancers, because there's some tricks we can do with these things, I have to say, that again could never be done before. If you can imagine now, we figured out how to send little old nanobots, if you want to call it, off to the lymph nodes. Well, we can pretty well figure out how to send them off to a tumor also. So I mean, it's where one wants to send these things. So that's where I'm very excited about going forward. So and I think hopefully we'll solve that and bring that to clinical, sometime to clinical endpoint before I retire to grow roses again, which I've tried to do several times, I have to say.
Oh, very
But
nice.
yeah,
Well,
no, yeah.
Dr. Thomas Rademacher, thank you so much for being on the Life Science Success podcast. I really appreciate you being here and telling me about Emergex and everything that you're working on. I really appreciated kind of the journey of your career and everything, that you're working on as well. I mean, it sounds extremely foundational, and I greatly appreciate your time.
Okay, thank you very much.
Thank you for listening to Life Science Success. For complete details about this podcast, including show notes, how to get in touch with guests, and more episodes, please visit www.lifesciencesuccess.com. If there's someone you'd like for us to invite to the show as a guest, please let me know by sending me a message at the podcast website. Please click subscribe on your favorite podcast app, share the podcast, or tell a friend about it, and last but not least, rate the podcast. Thank you again.